Please welcome our next discussion, Prevention, Prediction, Precision. A conversation between Lori Glibcher, President and Chief Executive Officer for the Dana-Farber Cancer Institute, and Cyriac Roding, Co-founder and Chief Executive Officer for Early.
So welcome everybody. It's great to be here and with you, Cyriac, to talk about cancer detection and prevention and early risk and detecting early risk. I thought I would ask you, I've heard about your company Early, and of course, we all owe a huge debt of gratitude to Dr. Sam Gambier, who unfortunately passed away a year ago. And I know from talking with you before that he was one of the real inventors of Early and was really dedicated to moving cancer back so that we could detect it as early as possible. And I really enjoyed the article in The Atlantic. So I was hoping you could tell us more about Early and Dr. Gambier.
Well, it's great to be here, Lori, with you and with your very special vantage point, Dana-Farber. You see a lot of different things, so it's going to be an interesting conversation about Sam and about Early. First of all, Sam is just a very brilliant, or was a very brilliant scientist, and surprisingly an even more brilliant human being, a very special man who came up with the concept of Early, sort of born out of frustration with the pace of progress, because he said we're racing against the clock. Every day that we're not making massive progress, we're losing people from cancer. We need to find this disease earlier, because the chances of survival are between 5 to 15 times higher, depending on the indication, if you find it early at stage 1 versus at stage 3 or 4. So he then said, okay, liquid biopsies work by looking for naturally shed biomarkers in the bloodstream, and you're taking an 8 milliliter sample out of a 5 liter total blood volume. The earlier you get, the less material there will be, right? And if you look at the liquid biopsies today, detection rates for stage 2 are somewhere around like 30 to 40%. Stage 1 is like 10 to 20%. So that's where Early came in, because Sam said, well, what if we could flip this on its head? And instead of searching for something that's naturally shed in a small tube, what if we actually went into the body and we forced the cancer to make a non-human synthetic biomarker that doesn't exist in a human body? For example, limonene. Limonene is in oranges, not in humans, unless you eat them. And if you haven't eaten an orange, but you're exhaling limonene, you might have cancer, because the cancer was forced to make it. So now the second step kicks in. You're forcing the cancer this time to express an enzyme that eats up a radioactive PET tracer that has been injected. And that PET tracer is now getting stuck in the tumor and makes a tumor that's normally too small to image, imageable. And now that it's imageable, it's localizable. And now you can treat it with precision surgery or targeted radiation. And in the future, you might even be able to use the Early platform to trigger a localized immune response in the cancer and force it to kill itself. So I'm going to stop here. So that's the Early story, and that's what we're working on. It's a moonshot, as you can tell, and there's a long way to go, but we started it.
Well, Sam made some fantastic creations already, right?
Yes.
He made a smart toilet that analyzes urine and stool to detect biomarkers of colorectal cancer, which could allow us not to have colonoscopies. Wouldn't that be nice?
That would be very nice.
A photoacoustic smart bra that enabled doctors to hear light reflected by blood vessels that form around a microscopic breast tumor. And then there are sensors that can register smell prints of lung and ovarian cancer. One of my wishes, if I could do this, is to create a dog's nose in the form of a chip, because we all know that dogs, particularly dogs like Labrador species, can sniff out the urine and identify cancer. And I remember one study that was done a while ago of that trained the dogs, and they gave them 100 samples of urine.
Yes.
From normal people and from patients with kidney cancer. And those dogs had close to 100% specificity and sensitivity to those. They identified all 100 of the patients. And then some dogs kept on sniffing one of the so-called control urines. And finally, the physician, the primary care doc of that particular individual, did a kidney MRI and found a small tumor. Really remarkable, and I don't see why we can't someday convert that into an electronic nose, because dogs have an exquisite sense of smell, because they have, multiple more, thousands more olfactory receptors than we do.
Absolutely. In fact, Sam's Canary Center for Early Cancer Detection at Stanford is working on an olfactory approach as well. I'm not sure what the latest is there, but you are absolutely right. I think I would love to know from you as well, like at Dana-Farber, you have this extraordinary vantage point, because you're seeing so many different disciplines come together. You see scientists, you see physicians, you see multiple indications, you see what is being done in the clinic today, you see what's being worked on. What do you see, Lori, as the major trends, the major opportunities, and the major obstacles on the course to prevention, prediction, and precision?
So this is a top priority for us at Dana-Farber, because as you pointed out, if you can detect a cancer at stage 1, you're highly likely to cure it. We don't do as well when the cancer is already spread. And that's true of 75% of the patients we see. So, I kind of divide this into identifying high-risk individuals for either because they have family inheritance. 10% of cancers actually are familial. They get passed along in the germline. And there's no reason, no excuse for why that 10% of patients don't get detected very, very early. And so we've set up a number of things at Dana-Farber. For example, Dr. Judy Garber, who is our breast oncologist, has raised enough funds to sequence all Ashkenazi Jewish women for BRCA1 or other mutations that are known to be present and that are passed down.
Yeah. Because
those patients should be guarded carefully. And we also have founded the Lynch Center. Lynch disease, Lynch syndrome, named after Henry Lynch, right, is about 5 different mutations, and that leads to a high risk of colorectal cancer and uterine cancer and pancreatic cancer. So every time we see a patient with colorectal cancer, we sequence the genome to see if they have one of those mutations. And if they do, we bring in the whole family. It's a program that's Dr. Sapna Singhal produced at Dana-Farber. The point is that we interrogate all family members, and not just, nuclear family. We get the cousins, et cetera, because you have to have a colonoscopy more often if you have a mutation, and nobody likes having colonoscopies, but it's a heck of a lot better than presenting with metastatic colorectal cancer. And that's a disease that I'm particularly concerned about, because there has been an alarming incidence of colorectal cancer in young people. I have seen several individuals now that are treated at Dana-Farber. They're in their 30s, they're in their early 40s, they're healthy, they have no comorbidities. There's been a recent surge now in young women, particularly in their 30s and 40s, who are non-smokers, have never smoked, and who are developing lung cancer.
So what's happening? What do you think?
I think that this points out why fundamental research is so important, because we don't know why. Could it be obesity? In some cases, we know that obesity is going to pretty soon supplant tobacco as being the major lifestyle behavior that leads and increases your risk of cancer, whether it's lung cancer, colorectal cancer. And how does obesity do that? Probably through the inflammation that gets activated in fatty tissues.
Right.
So I think that's one possibility, that that obesity, and we know that's true. We know that women who are obese have an increased risk of developing breast cancer and a poor survival outcome. I would say that we need to figure out the molecular basis of why obesity leads to inflammation and why inflammation leads to an increased risk for cancer. We know that's true. And so here you have fundamental research interacting with clinical trials to test the effect of exercise on breast cancer and other cancers.
Prognosis seems to be as important as diagnosis, especially in early detection, right? Because otherwise we're going to have lots of diagnosed people who actually don't need treatment. So how well do the new technologies that you're seeing, the early detection methods, perform on that front? Liquid biopsies, other technologies.
Yeah, I mean, I think that there's another approach as well. So there are diseases that declare themselves at very early stages. And then one good example of that is multiple myeloma, which is increasing in incidence, actually. And that is often preceded by what we call monoclonal gammopathy of unknown significance. And we used to wait until somebody had, obvious multiple myeloma to start treating them. And that just doesn't make sense, right? And so Dr. Irene Gobriel, the Dana-Farber, has set up a clinic because early detection gives us so many potential opportunities. What Dr. Gobriel is doing is she's looking at the precursor conditions of myeloma, which is, as I said, monoclonal gammopathy of undetermined significance, and also smoldering multiple myeloma. So she initiated the Center for Prevention of Progression of Blood Cancers, we call it CPOP, where patients who have precursor conditions such as MGUS, or early myelodysplastic syndromes, or early chronic lymphocytic leukemia are monitored prospectively, so that when we see any clonal evolution or disease progression, we can now treat those patients early. And the earlier, it's not different from some of the inflammatory diseases like rheumatoid arthritis, where the earlier you treat a patient with TNF blockers or IL-6 blockers, the better off they do. And the same thing can be said for cancers.
How far away do you think we are, Lori, from a world where diagnostics, or better prognostics, I should say, together with therapeutics come all in one? So, for example, you have something detect that there's a problem, and then in a next step, it automatically activates the immune system to take care of it and get rid of it. Theranostics is the key word here.
Yes, and I'm a big fan of theranostics, and it's one of our priorities at Dana-Farber. But when you're looking, if your readout is activating the immune system, and it has to be activating the immune system selectively to a given cancer protein, right? Because the immune system becomes activated all the time.
Right.
So, you're going to detect whether somebody has a cold or a virus, or whether they, have a cut and it's being scarred by appropriate immune cells. So that's a tough one, but theranostics to me is really amazing. The ability to diagnose and treat with huge sensitivity so that you can get down to the sensitivity of one cancer cell,
right,
with imaging, and you can then at the same time kill that cancer cell,
right,
is enormously exciting. And this to me is the future, is one of the future prospects that are going to really allow us to detect cancer at very, very early stages.
Now we're at full circle with Sam Gambier's vision, right?
Yes, absolutely, with Sam's vision. And so we have some scientists working on theranostics at Dana-Farber, and the first approved diagnostic theranostic should be on the market in a few months for prostate cancer. And we have set up treatment rooms, so that we can accommodate all prostate cancer patients who want to be examined, and then we can use theranostics, diagnostics, and therapeutics.
Yeah.
Which is wonderful for the patient. It limits the destruction of normal tissue and unfortunate side effects from prostate cancer surgery that can occur. So we're very excited about pursuing that at Dana-Farber.
Yeah, the thing that really drives us is the idea of, well, you know, if early cancer detection becomes better and better, then at some point we're all going to face the problem we already have in the clinic today. In lung cancer, for example, you know, 94% of all detected lung nodules are benign, 6% are malignant. The problem is no one knows which 6%. So it would be critical to ask the question, what if cancer detected early could actually be acted on? Because unless you know where it is inside, not just which organ it is, which tissue it is, but also where in the tissue, it's really hard to act on it. So that is what drives us and keeps us awake at night, because we want to find out how we can help all these patients that are being diagnosed.
Yes, it is very difficult for our oncologists. And that's why we have to develop enormous sensitivity and specificity in any of these detection modes. And I think what you're doing it early is incredibly innovative and exciting. And I think that theranostics will be a field that will continue to expand as we realize that, you know, you could go in there if you have a very, very tiny lesion. Not a big deal if you can use theranostics to obliterate that small nodule, whether it's cancerous or not.
Yes, I can't wait for the day when this becomes real. That would be my dream.
I would say also that, you know, the future includes prevention, right? It includes cancer vaccines, which mostly to date have not been effective.
Correct.
But we actually have a very strong group of scientists led by Dr. Kathy Wu, who are making therapeutic cancer vaccines, which appear to be effective in melanoma. And a German group confirmed those data. And Dr. Wu now has potential cancer vaccines for kidney cancer, for ovarian cancer, for glioblastoma that are in clinical trials now. So that's therapeutic vaccines. But of course, what do we look forward for our grandchildren and great-grandchildren? We want preventive cancer vaccines. We want to know enough about the individual and both unique and common proteins that sit on the surface of only cancer cells, not normal cells, and design vaccines against those peptides, so that one day our kids will get mumps vaccines and measles vaccines, and they'll get their cancer vaccines that will cover a large portion of all of the neoantigens that are present on tumor cells.
It's very important. I think we are all coming from a world where medicine used to treat people who are already sick, but what if we could keep people from becoming sick or intervene really early? And that needs to be, evolved also on the healthcare and reimbursement level.
It does, and it requires a great deal of collaboration between fundamental scientists, clinical researchers, clinical trialists, and industry. We want to get treatments into our patients as soon as possible. We want to take our fundamental discoveries and turn them into drugs. And the best place to do that is not necessarily in academics, although we do actually have a great chemical biology group at Dana-Farber, but most of the time you want to hook up and work with a biotech company or a pharmaceutical company to move that forward as fast as possible, to get it into patients, translate what we found. Dr. Bill Kalin at Dana-Farber, who won the Nobel Prize a year ago, started out with a very fundamental question, how do cells sense oxygen? And he pursued that for 20 years, more than that. And now there are drugs based on his discoveries of how cells sense oxygen on the market for anemia and for kidney cancer. And he didn't do that all by himself. That was work done in collaboration with the private sector.
The best innovations often happen on the intersection between worlds that normally don't talk. And you can see that between academics and industry. You can also see it now in the type of biology work that's being done. Biology used to mean wet lab only. Now it means a combination of wet lab meets dry lab, and bioinformatics meets pipettes, and bits meet cells, and physical worlds meet digital worlds. And when you bring those together, engineering, biology, and computer science, you get some enormously new innovative approaches that haven't existed before. And these intersections are where major innovations are going to happen on the genomic side as well.
Well, I would say that the future of cancer care absolutely relies on leveraging more technology, and that includes massive buckets of data, artificial intelligence, machine learning. And my dream is that 3 to 5 years from now, when a patient first presents at Dana-Farber, and we gather what I'm calling like 6 buckets of data, and that ranges from the genomic profile of the patient, the immunoprofile of the patient, pathology, radiology and imaging results, microbiome, metabolism. If you can analyze that, collect the data and analyze that, if you do it with enough patients, you probably need hundreds of thousands. I think we'll be able to tell a patient when they first walk in the door and after they've had their analysis done, what drugs are going to work for them based on how other people have, with the same profiles, have done on these drugs. We're going to be able to say this drug is going to probably work for your cancer, but this drug probably isn't going to work for your cancer based on all the knowledge we've accumulated. So what is the future for early? What do you think the timeline is, Cyriac?
Well, I'd like to have it out yesterday.
Yeah.
But the current situation is we moved from in 2.5 years since we started the company, we moved from mouse data to canine data, went from a 30-gram mouse to a 15 to 35 kilogram dog. That's a factor of weight of 300 to 1,000x. Going to humans is a smaller jump then of 2 to 9x from dogs, also because they have spontaneous cancers. If all goes well, knock on wood, we will be starting our first human trials in Australia in the next month or two in late stage lung cancer patients, and then going to earlier stage right after that. And then we will bring the data back to the US and to other markets after that. So I'm hoping that in 2 years from now, we will be getting to a phase 2, and then in 25, 26, we'll be ready to go into the market and commercialize. We are very, very focused. We want this to be helping patients. I'm not so interested in just constantly doing things in the lab. We want to get this out of the lab and into the clinics, my wonderful day will happen when we have the first patient who we have helped find cancer early and treat it early, so they can move on to another chapter of their lives and think about this as a history that happened to them, but then move on with their lives and live a good life after that. That is the future that I'm dreaming of.
Sounds good to me.
Okay, and at Dana-Farber, I think there are so many great initiatives going on as well. I think we heard a lot about this today, and I'm fascinated by the work you guys are doing. Thank you.
Well, thank you for keeping Sam Gambier alive in what you've done at early. I'm sure that if he were here, he would be thrilled with the approach that you're taking, and it's an exciting one, and high risk, high reward.
Yes, no risk, no return, and moonshots need to happen in order to get to Mars. So we'll see where we end up in the end. Sam is very much alive in our hearts, and I thank you for saying those kind words.
I think we have a common purpose and mission ahead of us.